Welcome, everyone. Today, I'm really pleased to welcome Professor Sean Schoville from our own Department of Entomology. And yeah, over there, actually, in addition to his research and teaching and mentoring roles, he serves as the chair of that department and also the director of graduate studies for the brand new Ecology and Evolutionary Biology program. So has a little bit on his plate, plus I would also be, remiss if I failed to point out that he has won major awards here for both his mentorship and his teaching. And then there's his entire research program, which, as we'll hear about today, does involve genetic adaptation of insect crop pests. But also involves a whole range of other systems, both things of biomedical interest like ticks, and also things of significant conservation interest and ecological interest, such as high altitude insect species of various types. And a lot of these insects don't necessarily have strong genomic resources to begin with, so those often need to be established along the way. So yeah, so today, Sean is going to be going to have to select, one interesting part of that broader research program to tell us about. And so I'm looking forward to hearing about it. So thanks for being here, Sean.
Thank you, John. Thanks, folks. Really a nice opportunity for me to talk to you. I appreciate that. So thank you for coming. I haven't been able to talk to genetics before, so this is great. I'm really looking forward to talking with you. I do want to apologize right at the front for the students that were here. I was supposed to stay an hour afterwards, and I flipped on that because I have another engagement. So my apologies. I did get your questions, and I'm going to try to do my best to weave in answers to some of those questions in my talk. But I will be here for a question period, so please just feel free to ask those questions again. This is the Colorado potato beetle. What I want to start with is a story about it, and really, oh no, this isn't working. A story about it, but how it became a problem. So this really starts with potato itself. As you may know, potatoes come from South America. In Peru, the high Andes, Bolivia and Peru. And they were discovered by Europeans when Pizarro came conquering into South America. Among other things, he took potatoes back with him. And so potatoes spread throughout Western Europe and then eventually the rest of the world. And why is that important? What you may not realize is how critical potatoes were at a time when the human population was growing rapidly, but there was a repeated famine. So this is just for France, but this chart shows you a series of famines between 1250 and 2017 of severe famines that led to, significant population impacts. And those were occurring because the crops that people were depending on primarily were forage crops like wheat, and those don't always ripen correctly, especially if you get really wet conditions. And so there would be crop failures and there would be mass starvation. So potatoes get brought to Europe, and that's by the end of the 18th century, many people are eating potatoes, and to such an extent that about 10 to 30% of the population ate only potatoes as their principal food source. So it's a very significant change in diet, and it's really the population depended on it. And so it's in this context that we then get a horrible pest that starts to decimate potato, and that's why it becomes particularly important. I'll just say that as part of this story, you may have heard of the potato blight. This is a fungal disease that affects potato. Of course, there's a famous Irish potato blight, devastated potato production in Ireland in about the 1840s and led to a mass migration. And so as folks left those conditions in Europe, they often came to the United States. And as they came to the United States, they moved west, developing homesteads. And this is a story about how the potato beetle became a pest. So this is a map up here that shows you through time the potato production in red. It's rotating forward in time. It'll recycle itself. But what you see is that potato is grown along the East Coast, and it begins to spread westward in the 1800s as homesteaders move west. So among the things they're growing wheat, of course, some maybe adopting corn, but they're also still depending on potato because it's such a reliable crop. And they bring it right west into the plains region where they find the potato beetle. The potato beetle is this innocuous little guy that's living on these weedy plants called buffalo burr, that if it stayed on those, we'd never have thought about it too much. But it does a host shift, and it quickly establishes on potato, and then it explodes. And then it moves rapidly eastward. So these are some dates here that you can see. The first outbreak is in 1859, somewhere in Nebraska. Within just a few years, it's already in Iowa. A few years more, it's Wisconsin. A few years more, it's in Long Island, New York. We think that it came over on the rail tracks because there's descriptions of bugs swarming on the rail tracks and the rail cars literally slipping on the masses of beetles that were pouring out of the rail cars. So what they would do is they'd load potatoes onto those rail cars from the west, send them back to the population centers on the east. And this is how the potato beetle starts to get moved across the country in mass numbers. I have this little quote here. This is a historical quote. I don't know if it's easy to read, so I'll just read a little bit of it. A day or two, a party of gentlemen fishing near the middle of Long Island Sound saw great quantities of potato bugs covering the surface of the water as far as the eye could reach. Every floating article, as well as the water, was packed with them, and they were clinging to eelgrass and seaweed under the water. The wind was blowing and blowing them from the island, and they were being wafted into the Connecticut shore. They appear to be increasing in numbers, and they started to attack eggplants, pepper plants, and tomato vines. These are all plant species in the Solanaceous family that are very close to potato. So just this massive explosion of these insects attacking a kind of a vital food crop. And the note I put here about the Long Island Sound is telling in the context of this video, so I'll play this. This is made in Denmark and kind of features how the people there were dealing with the potato beetle kind of in the early 1900s. So important crop, a food staple that's brought to their table, ooh, lovely potatoes. And then it'll segue in talking about the potato beetle and its biology, describing how it eats potato plants, lays eggs on those plants. Those are some little aphids crawling around the eggs. These are the larvae. And then it'll show you some images of them defoliating the plants. So they chew the leaves, and they can actually attack the stems as well. And eventually, they will just literally defoliate the entire plant. So they leave the tubers under the ground. Those aren't affected by the potato beetle. But because of the loss of the plant material, the tubers don't grow very well and it's not very productive. So that it leads to these dramatic yield losses. So this is a defoliated patch. It's kind of black and white, so it's hard to see. But of course, there's ways to address this. This is early attempts to spray insecticides to treat them. You can see at this time they didn't use protective gear. They're spraying pretty toxic stuff. This is another fellow doing hand spraying. And you watch it blow back into his face. It's really great. And then what they did is they tasked children to go out and manually pluck potato beetles from the plants. So this is along the shoreline because those beetles that were washing out into Long Island Sound were crawling on ships. And then they were also washing across the Atlantic. They can survive in saltwater for weeks at a time. And so they were finding them on the seashore, and they would eradicate them with very dramatic measures, right? The picture of the kids plucking them by hand, this was a common thing in Europe. My mother did this as a child. She's French. So they were tasked by their grandparents to go out and pick the potato bugs off the plants. So really, it's like this very significant issue for people. Okay, so this is where insecticides come in. Manually taking care of potato beetles is laborious, doesn't always work. So quickly, there was an interest in developing chemical control. The first chemicals used, 1867, are Paris green. Paris green is something that was actually used for green paints. So Van Gogh's famous self-portrait in green there, that's Paris green that's used as a pigment for that paint. But they realized it was poisonous and toxic, anecdotally why perhaps Vincent and others suffered various conditions over time, nervous conditions. So Paris green was known to be used effective in killing insects. So they would deploy this and spray it on the potato plants. They also used lead arsenate and other compounds at the same time. And then later we have, of course, the development of industrialized insecticides like DDT. They came out of the war effort in Germany and other places. DDT was developed in 1939 and then quickly deployed in Swiss potato fields specifically to test on potato beetles. It became available for use in 1945, and potato beetles developed resistance to it in at least in one place in 1952. So this is the premise of my story, is that we have this horrible thing that's affecting our food. Food's important, and we quickly developed strategies to deal with it using chemistry, but very quickly there's resistance. Just some other fun anecdotes. So in the war, after the war, there was a lot of propaganda that the US government was using potato beetles as bioweapons, that we would drop them from airplanes in East Germany and Berlin. And so these are propaganda posters from East Berlin about those terrible Americans doing this. And we don't really have a record of Americans actually doing this with planes. The beetle was spreading well enough on its own throughout Europe. It moved in through Russia. It eventually got to China. So it's very capable of spreading quite on its own. And then this is another picture, and it's showing you some kids that were in a contest to collect potato beetles. They had contests. And so this young fellow collected 2,000 in one day and won the competition. So just kind of fun anecdotal stuff. Okay, so the take-home message I want you to have is that these pests, like potato beetle, and we can think there's many other species that we're now working on, are really important problem. They're a financial problem that we address every year. So it's billions of dollars that we spend on pesticides, on the fuel to spray pesticides, on the labor that's involved. Plus it has all those negative downstream effects in the environment and all the remediation that we try to do. So it's a very expensive procedure. But if we weren't to do it, what we currently experience is about 35% loss in crop yield to pests. And that would be expected to double if we didn't have pesticides. So we'd all like to live in a world without those, but we really need strategies, alternative strategies to address this. Otherwise, we're going to be facing pretty significant consequences in terms of food production. And looking forward in time, we know with the population growing that the food demand is going up. So it's not really like the problem's getting any easier. We seem to be able to produce enough food for the world at this point, but with those increasing challenges, this is becoming more and more of an important issue. So there are a lot of pests to choose from. I told you a fun story about potato beetles and why they're kind of charismatic. But there's other couple of reasons why I picked them in particular. I didn't work on agricultural pests before I came to UW-Madison. I really started that here because it was a strength in my department. I could collaborate. I also saw the opportunity to get support from different industries, like the state potato growers support my work financially. So it was an opportunity for me. And as I was thinking about pests to study, I was thinking as a geneticist about what the properties were that I was most interested in. One of them is that potato beetles don't disperse very well. So that means that genetic structure emerges on the landscape. Genetic diversity is sort of partitioned across space. And you can actually make comparisons across space to learn something about how they're adapting. So that was important. Other pests are like aphids, and some things are windblown, and they can move thousands of miles. And so they're effectively one single panmictic population. And so it's very hard to use spatial sampling to study their genetic variation and connect it to adaptation. But there are other reasons, and I told you about resistance evolution. The potato beetle is one of the most notorious pests in terms of resistance evolution. It's evolved resistance to over 50 different compounds, where those compounds are attacking the pest at different places in its nervous system or other parts of its body. And so that's really significant. Like, it's not just resistant to one class of chemicals, but a whole bunch of them. This little figure shows you kind of the development of different chemicals through time. They're colored by mode of action, which is how it attacks the insect at the cellular level or in the nervous system, for example. And so you can see that the colors change because there's new chemistries that get developed over time. And what the graph shows you is the year of introduction on the x-axis, and then on the y-axis, how long it took before resistance was reported for Colorado potato beetle. And if you just imagine a line in that, you'll see it's quickly trending downwards, so that recently in time, the rate of evolution to develop resistance has accelerated. It's very fast now, happening at a much quicker time scale than it did historically. So it's not just that they're resistant to multiple things, but they seem to rapidly evolve resistance as we introduce new chemicals. And that's kind of interesting. And so what I'd like to do is think about the evolutionary mechanisms that are linked to this, how we can explain those. Also the genes that are a part of that story. So what specific genes are changing to deal with, to develop that resistance. And so that's kind of the heart of my talk, is to walk through that. I mentioned rapid evolution, and for a moment, I'll just pause to say what I mean by that. It's kind of this generic term, oh, rapidly evolving. You can kind of throw it out there and it can mean a lot of things, but actually has been defined. And this is where I went to Hairston et al. In 2005 to get this definition. And if we think about rapid evolution, all species evolve, and some can do it quickly in real time, in human time, but that's not really what it is. It's about a rapid rate of phenotypic change relative to generation time. So we measure phenotypic change in terms of Haldanes. So that's like the per generation change in phenotype relative to the variance in that trait. And we can do it in generation times, and this is showing you log generation times. And so this is the figure from Hairston et al. And what they did was develop some evidence for rapid evolution and defined it as those cases that were occurring, in log 1 or less of generation time. So where do the potato beetles fall in this kind of plot? Well, most insects would be out here in terms of insecticide resistance. We did a survey of the reported time to first case of resistance in this paper with Christian Breivik. And so it's usually about 66 generations, and this is across 20 major species. So that's the average. It's not particularly rapid. Potato beetle, the fastest record is this. It's right at the front end of that. So in one generation time, a population in New York developed resistance to a new insecticide, a neonicotinoid that was introduced. And so that's the example there. And you can see that's pretty exceptional, right? So that's sort of an interest in and of itself, is to try to understand what's causing that phenomenon. I also want to frame this kind of in a little bit of a fun way for the genetics audience about the historical debate that's been had in the literature about how adaptation occurs. Hopefully you all recognize James Crow there, lecturing here at UW-Madison in the 70s. He famously did this study on Drosophila melanogaster that was DDT resistant. He was interested in that problem. And what he did is he brought field populations into the lab and did lab selection to see how long it took to get DDT resistance and how that DDT resistance sort of evolved. And what he found, or what he deduced from his work, was that it was likely to be a polygenic response, that there must be many genes involved that pre-exist in those populations in the field. And as you select for them in the lab, that develops the insecticide resistance. So this is early work that shows that this concept of that standing variation, that variation out there naturally in populations, particularly in like a polygenic model, could be harnessed to evolve under strong selection in the wild in insects, particularly to insecticide resistance. I'll just say that was there and somehow got looked over or ignored for 40 years basically, because most people fall into this camp on the other side, which is believing that insecticide resistance evolves as a single strong mutational effect, a single gene, but basically. So often even they claim that it's likely to be recessive because of the fitness trade-offs that are involved. And that if we plan our strategies of management around that model, we can delay the onset of resistance evolution. So folks like Bruce Tabashnik and others have developed these pretty expansive models, mathematical models, to explore how to manage pests to avoid the development of resistance. And the basic premise of one of their more famous strategies, a refuse strategy, is that if we maintain susceptible populations adjacent to where we're doing our selection, that the gene flow will counteract the evolution of the resistance gene, and so swamp it effectively. So that's sort of the basis of a lot of management and what we call integrated pest management in agricultural systems. And most folks have built their plans around pesticide use, coordinating responses on landscapes with this model in mind, and really overlooked this other possibility that selection could occur through polygenic mechanisms, and that it could just be there already in this standing variation. Okay. How do insects develop resistance? There's a number of mechanisms. Many insecticides are targeting the nervous system, for example. And so if you develop a mutation at the target site of where that active compound is attacking the insect, you might develop resistance. So we call those target site resistance mechanisms. You can see a few of them up there. So those would typically be hard genetic changes with strong effects. Those are the things we expect to be strong effect mutations. There's other kinds of resistance. Penetration resistance refers to thickening of the cuticle or some alterations in the chemistry of the cuticle to limit insecticide sprays from penetrating into the body. Of course, microbes in the microbiome could be a component of insecticide resistance. We know that microbes develop insecticide resistance, so they could be conferring some advantage to the insect host. And what I'm particularly interested in is this metabolic resistance. This is a type of resistance where the insects detoxify the chemicals. So they break down the chemical in a series of steps and eventually excrete them from their system. And what's interesting about that is it's a more general way to deal with a whole range of insecticides, not necessarily a specific insecticide. So this could be a strategy that allows them to be resistant to multiple kinds of insecticides pretty quickly. This is just a kind of a snapshot of what target site resistance might look like. So there's a whole lot of different insecticides. They attack different things, acetylcholine esterase receptors and sodium channels and all sorts of things. And so particularly they've been designed to have strong effect in the nervous system of the insect. And so we can imagine the resistance being a mutation at those receptors or at the at some sort of step in that process of signaling at the nervous system level. Metabolic resistance could come about in a number of different ways. Because it involves many genes, there could, of course, be some protein coding mutations, things like that. But we often expect gene regulatory changes to be important here. We might also expect things like gene duplication events that rapidly increase the expression of certain genes through duplication to be important. We often, there could be cis effects, trans effects, and so forth that might drive whole pathways. So as it happens, many detoxification pathways have upstream transcriptional factors that regulate the whole downstream series of steps. So some rather simple changes could actually be consequential in driving the whole detoxification pathway. And I'll just kind of also point out, like, there's that view of fitness that was tied into how we think about resistance evolving. Traditionally, they viewed this trade-off occurring. If insects were going to become resistant to DDT, they had to give up something in order to do that. And they would have low fecundity or low survivorship or something. In fact, we rarely see that. So when it's actually been measured and studied, there's rarely trade-offs for the evolution of resistance. What we typically see is sort of delays in longevity or growth. So they're not necessarily extremely consequential. So I think that just suggests that our imagination about how to manage these problems in nature needs a little broadening, needs a little bit deeper thinking about how these mutations come about and what their consequences are. Okay, a little background on how we're going to look at patterns in genetic variation. There are a couple of frameworks that we think about when we think about looking for signatures of selection in the genome. The simplest one is sort of shown in this plot on the top. When we have a new mutation arise in a population, and that's favorable, we quickly see that it becomes selected upon and fixed over a series of time. And that is what we call a classic hard sweep. That hard sweep erodes nucleotide variation around it. It carries a specific haplotype to high frequency. And so nearby variation gets diminished. And so if you scan across the genome and look at nucleotide diversity, you'll see a deficit where there might have been a selection event. You also typically see some strong evidence of linkage disequilibrium in that local region. So these are some nice ways we can kind of scan genomes and look for selection. Another model, and I talked about standing variation, suggests that you wouldn't likely see as strong of an effect. If there's variation that's already there, maybe on multiple haplotypes, both those multiple haplotypes might equally be selected. So you don't see this diminishment of nucleotide variation adjacent to the selected site. And so you have more of a soft sweep, it's called. Those are much harder to differentiate statistically from background genomic variation. But there's been some development of tools and strategies, in particular, often comparing populations and things like this can help you start to identify some of these soft sweeps. Those are two important things. The last thing I'll mention about the genetics of pests in general, these beetles, is that there's a lot of redundancy in their genome. So this is another form of standing variation. Often they have, like in the detoxification family of genes, there's many, many genes. I forget exactly how many ABC transporters there are in the Colorado potato beetle, but it's like 90 or 100. And those are key genes that are involved in the last step of detoxification, the excretion step. So because there's so many of these genes, in a sense, they act as multiple templates upon which a mutation can occur, and they increase the probability of there being a beneficial mutation, right? So that redundancy across the genome is another form of standing variation. It's not just polymorphic sites at a particular gene, it's also related genes that could have similar functions. Okay, so what I want to do now is kind of walk through the work we've been doing to try to understand how the adaptation takes place in Colorado potato beetle. Some of the first questions we had were about the history of the beetle, its evolutionary history, and trying to refine that. There were some ideas of what had happened. We wanted to test those with genetic data. In particular, I was interested in the demographic effects of becoming a pest and how that was going to then impact my ability to screen genetic variability to look for adaptation. So if there's genetic bottlenecks and things like that, that can really erode genetic diversity. So we want to know about that if we're going to go on and test for adaptation. So one of the first projects we did as I got here was to screen 85 individuals with whole genome data. This time this was quite expensive. And so we were pretty limited in sample size. We sampled them from across the United States in a couple of key regions where they're pests and other places where they're present on potato, but they're not particularly pesty. So in particular, this Pacific Northwest region, Washington, Oregon, and part of Idaho, they occur there, but they don't really bother potatoes too much, and they're well controlled. Of course, that's where most of the potatoes are grown, so that's a good thing. But in the Midwest and in the East Coast, they're really problematic, and they have a lot of resistance. So we sampled intentionally to sort of compare those populations. We also sampled through the central part of the US there, the Plains region, because that's where we think they came from ancestrally. And so the genome's about 800 megabases, is a pretty normal size for a beetle. And we found a lot of variations, so 76 million SNPs. And this plot on the bottom just kind of shows you a representation of their structure, their genetic structure. And so we see some genetic structuring. The populations along the East Coast are all sort of forming one group. Populations in the Midwest form one or two groups. That Plains region is pretty distinct and has a lot of variation. And then that upper Pacific Northwest is distinctive. So there's some genetic distinction here, some underlying differences in their background variation that's important to track. We looked into signatures that there might have undergone a bottleneck as they rapidly expanded eastward. A very simple way to measure that is just to count singleton variants. You'd expect that when a population goes through a bottleneck, that some of those rare alleles, those singleton alleles, get lost because they're so rare in the population. So this first plot kind of shows you the patterns of variation. This box at the top that's has a lot of singletons. That's the Plains region where they're from ancestrally. So we expect them to have the most singletons, the most background variation. And then as we move to the left of the plot on the top, those are Midwest, Northwest, Northeast populations, kind of all the samples combined together. And you can see there's been this reduction. So in line with them moving across the United States in a spatial expansion, fewer, not the whole population went. So there's essentially a small bottleneck that erodes some of that variation and allows those populations to establish in the potato growing regions. Yeah, that's basically what I wanted to say about that. We used some methods to try to estimate the timing of this, and very nicely, it actually lines up with those historical records. So this is a approach of using the allele frequency spectrum, the site frequency spectrum, jointly between two populations. And you can use that information to estimate time of divergence, as well as the population size in the present day populations and the ancestral populations. And what we did is we sampled in Wisconsin and Long Island. We knew the dates when they were first documented there. And we did whole genome sequencing in 53 beetles from these two places and reconstructed that history. We did assume a standard molecular clock that was taken from other insects. We don't know the mutation rate in potato beetles, so we assumed that it would be similar to other insects. But it works out pretty well, and so we see the timing line up pretty well with that historical expansion event. So we estimated a divergence time between these two populations of 1855, and they were established 1867, 1872. So it makes sense that they would have diverged just before that establishment. And you can see that relative to the ancestral population, they experienced some growth into the present. So that's to be expected as well. As they moved into potato, they did quite well and then rapidly grew as a population. So that's sort of signified there in those large effective sizes. So this is a bit of background that you have to use to interpret some of the genetic data, that the species underwent this spatial expansion, may have lost some of that standing genetic diversity, actually maintained a fair bit, but then went through these rapid population growth phases. So they reestablished some of that rare variation because of that rapid growth phase. So what we've done now is to look at selection kind of at a large regional scale, but also at a local scale. So there are two pictures here. On the left is the Wisconsin potato production region. I've sampled a bunch of populations in Central Sands, which is a 2-hour drive north of here. And what I draw your attention to, these are measurements of insecticide resistance to neonicotinoids. It's a commonly used insecticide. And if it's in black, they're more resistant. If it's in white, they're not very resistant. And what I would point out there is that the heterogeneity that you see in the phenotype of resistance is pretty impressive. So you go site to site, field to field, and you can have different patterns. I think this is pretty telling because if you thought that the resistance devolved from a single mutation and then spread from one site, this would not be the pattern you'd expect, right? You wouldn't expect there to be so much heterogeneity over space. You'd expect there to be like a wave of favorable, highly resistant populations kind of diminishing outwards. So I think just from the phenotypic data alone, we can tell that evolution is happening independently across this space, and populations are finding different solutions potentially to overcome the insecticide. At the larger scale, these are 4 regions kind of key interest for potato production. So this plot, the average relative resistance, is measured over a suite of insecticides. And it just shows you the kind of phenotypic performance of these different populations. In the Northeast and Long Island specifically, we have the highest average relative resistance in potato beetles. So this is a really interesting population. Is the one that developed resistance within a year. It has, in general, broad resistance to many insecticides. The Mid-Atlantic, the Great Lakes is elevated, but the Northwest is the least resistant relative to those. So a little bit of background on the phenotype. And then as we looked into the genomic variation, this is a busy slide, so don't pay attention to the table too much. But what we found is that there was really widespread evidence of adaptation. So we used different tests. We looked at SNPs and genes, using different comparisons, looking at specific pathways. The take home is that we found evidence for selection of many genes, and really in each of these populations, repeated evidence of genetic change. Often some of the same genes could be involved, but it wasn't the same mutations. So we saw this independent evolution of resistance, and particularly in many of the detoxification pathways. So some of that chart is breaking it out into different categories, like some of the target sites, as well as metabolic resistance. This picture shows you the ABC transporters and the numbers of mutational events on each population involving different ABC transporters. And you can just see that there's this wealth of signatures of selection. We also looked at for classic hard sweeps using scans of linkage disequilibrium and kind of focused in on two populations. So the Long Island population, kind of that key population, and then one in Wisconsin that we've studied a lot is kind of our go-to site in Wisconsin for high resistance. We use a method called RAISED, which is a like composite statistical test. It takes a site frequency spectrum also, as well as measures of linkage disequilibrium to get its power to detect hard sweeps. And you can see on the plot there, the Manhattan plot, the points that are really elevated above the baseline are areas that are under selection, under classic hard sweep expectations. And what we found is that there were really a fair number of genes in both populations, independently, 74 in Wisconsin, 89 in New York, only 9 of which were shared, right? So we're seeing selection, but it's often not necessarily involving the same genes in these comparisons. And in this set of resistant loci, it's a range of things, target site changes, as well as a number of metabolic genes in those detoxification pathways. This is a heat map that shows gene expression. And importantly, this is just of beetles that are unexposed to insecticides. So they've been brought into the lab, allowed to lay eggs. Those eggs have been reared up. And it's just constitutive gene expression, baseline expression that you sample in the larvae as they develop. And we compared different populations across the range. And I'm just showing you genes in the kind of classic insecticide resistance genes here and their expression patterns. And so what the colors are meant to show is the differences among regions. You can see on the top, they're grouped by color. Those are the locations they came from. And so there are blocks of overexpressed genes, constitutively overexpressed genes that differ across this geographical range. So each of the populations is finding a different set of solutions. They're all involving these insecticide resistance pathways, some of them metabolic pathways, but they're kind of doing that differently. Okay, so that's a whirlwind of sort of some results. And, you know, a fair question is, do you have really good evidence that these genes are under selection? You do these scans, you get these correlations. Can we trust those? And certainly one of our goals has always been to get to the functional level, to do some more further follow experiments. We've done a little bit of that using RNAi methods. We've proven that some of these genes are involved in resistance. But I think there's just so many that this is an untenable path. So there are different ways we can think about this. Another thing to consider is that I showed you the history of selection. And really, it's been this story of compound after compound being introduced and then selection occurring and resistance evolving. So when I say there are genes under selection, it's sort of what exactly am I talking about? Because as we think about the introduction of chemistries through time, we're layering selection event upon selection event upon selection event. So what I can look at now with present day samples is this hodgepodge of signatures of evolution to all sorts of things that happen at different times. And so I can say generically that there's polygenic resistance, but I can't say that that's the mechanism exactly of how a specific resistance to, say, organic chlorides occurred. So that's a difficult thing to do. So it's a bit of a conundrum. What's one to do when you have this sort of problem without going into a time machine? So we need a time machine. So we used a time machine. We went into museums and we went back. Fortunately, potato beetles were collected prior to industrial pesticide use. So we were able to sample them through these 3 time periods. And focusing mostly in Wisconsin and that Long Island area in New York. So this was really cool. And we went into Smithsonian, we went into the Field Museum and all these places to find these beetles. This is what they look like in their collections. What you can do is sort of provide a little moisture. You can stick them in like warm water and they're very dried on that pin and they sort of absorb that water very quickly and you can slide them off the pin. And you can drop them in a tube with lysis buffer and digest the DNA. That hole that's going through the body of the insect from the pin provides access to all that tissue that's inside. And you digest it in the lysis buffer. And then you can take that beetle back out and stick it back on the pen, stick it back in the museum, and the color doesn't even change. So it's really cool. We use this non-invasive approach to get the DNA. And we did that here in the Biotech Center. They had this archaeology lab that's was available to us so that we were minimizing contamination and trying to use best practices for working with archival DNA. You can see the locations of our samples. We couldn't really get them from just one site, so we had to sort of aggregate individuals across sites. In the end, we had 78 samples that we were able to sequence with whole genome sequencing, albeit at pretty low coverage. So that low coverage was a problem. We didn't get the quality of data I hoped, and I think if I could do it again, I would have invested more in sequencing, but it was expensive. But we got enough of it to basically do one test. And that test was to say, what are the allele frequency changes across time? Do we see strong shifts in allele frequency across the genome? How many genes do we see that for? And what we found was that we found a lot of genes, right? So in Wisconsin, there's 2,700. In New York, there's 1,600. Importantly, many of those are the same genes we already found evidence of selection for, just scanning present-day populations. So we support that result, that there's broadly been many genes involved in this history, this legacy of selection. Interestingly, the genes that we found in those two areas, 72% of the genes, not the mutations, but the genes themselves, are shared in both places. So we call this parallel evolution, that there's these common trajectories to a same set of problems that happen independently in populations because of underlying ancestral variation. So this is kind of a cool result. But what you'll see I didn't succeed in doing is trying to tie those genetic changes to specific time points in specific insecticides. So still we're at a loss there. We can't really say how evolution to organic chlorides occurred or neonicotinoids. We still have this problem that we know it's a layering of selection over time. So that's something to do maybe in the future, to go back to these museum samples and try again and do it with more care. What I've walked through is just a quick kind of showcase of the different projects we've done, really trying to understand the genetic basis of resistance, some of the mechanisms involved. We show that resistance evolves repeatedly in different populations. It has a lot of genes at play. It's polygenic. Particularly, we found a lot of evidence of metabolic detoxification genes being part of this story. That's important because metabolic resistance often means multiple resistance to many insecticides, or cross-resistance, if you will. And so as we go forward in time, we're going to be dealing with that kind of resistance trait. And so as we introduce new chemistries, we might be challenged. They may not work as effectively as they would work on a naive population. And to make that point clear, we've done some work. So on the left is a heat map of populations in Wisconsin, looking at resistance to 6 different insecticides. And you can see the correlation in resistance. So red is highly resistant, and there are certain populations that are highly resistant to all 8 insecticides. Others that have more mixed status, they're like in the southern Wisconsin, there's some compounds they're not resistant to. So this metabolic resistance seems to be present, at least in some populations, to some compounds. And then on the right, this is where it gets a little scary. The left is neonicotinoids, and the blue side is a new compound that's never been used against potato beetle. It's called plinazolin. It's now able to be used in Wisconsin, but it hasn't been used yet. And this analysis just looked at baseline susceptibility across a bunch of populations, including Wisconsin. And then we did a test to see if there was a correlated level of LD50. That's a measure of resistance in terms of dosage. And we do see a weak one. It's not quite significant, but there's a trend. And notably, like that New York population on the top seems to be more resistant to this naive chemical that's never experienced than other populations. So suggesting like there's some underlying metabolic resistance that could be important to it. Okay, what I'd like to do kind of with my last chunk of time is just talk a little bit about forward thinking, where we're going recently, and sort of what my vision is for improving pest management and harnessing genetic data in a kind of a creative way. So what I'd like to do, what I've been working on, is sampling temporally and spatially to track evolution in real time in some of these pests, to try to understand the factors that contribute to resistance evolution occurring in particular populations, and ultimately to develop tools, statistics, models, simulation tools that could help us make predictions to forecast where resistance might occur next. We've been measuring phenotypic data for neonicotinoids over a period of time since I got started. You can see on the left, this is a measure of mortality. So a higher mortality means they're less resistant, and lower mortality means they're very, very resistant. Over time, across populations of Wisconsin, they become more and more and more resistant. So we've tracked that through time, and this is a spatial map of that. We've gone back to the same fields or nearby fields to try to sample through time, and you can see the spatial pattern there. So we're trying to measure in space and through time how that resistance phenotype is evolving, and then along the way we're taking genetic samples, and we want to put this all together to try to understand how to forecast. In order to forecast, we have, of course, environmental data that could be help be predictive in how populations might evolve resistance because of the dependency of insects on growth conditions, like temperature conditions. We have that underlying genetic diversity, both standing diversity and maybe adaptive diversity, as well as we know from population genetics theory that population size is kind of a key component of the rate of adaptation. So we have measurements of abundance for these fields. And then we have information of land management. So how often is potato growing? Is there a lot of potato nearby? How is that potato rotated across time? This is actually a proxy for the intensity of management, the intensity and the amount of insecticides that's used. So that field management history is sort of a proxy for selection strength, right? So we can take all of these measurements. And so, for example, this is a track of abundance change through time in these fields. This is for almost a 20-year period, a little shy of that. And in red, you can see populations that have changed rapidly, grown rapidly. Blue ones have kind of stayed the same. And you can see that there's different areas where the population size is growing pretty strongly. They might have developed stronger resistance to the insecticides through time. And this is the model we put together. So we used an approach, it's a Bayesian approach called in-law, and it has some nice properties. I won't go into details, but one of the things that allows us to do is take a set of genetic samples and interpolate across space in a spatial way so that we're adjusting for spatial and temporal correlations. So we've done that to try, because we couldn't sample all these every single population of beetles. There's 1,800 fields, right? So we have a core set of populations that we've tracked through time with robust sample sizes. So I think in total about 465 beetles that are represented in the genomic data that are here, but it's only from 18 populations. And we've tried to interpolate across those populations and to their neighbors what their genetic patterns would be. So this method allows us to do that. So we put this together. These are the predictors in that model, and I've just boxed the ones that came out as statistically significant in terms of predicting insecticide resistance evolution, and that's the phenotype I'm talking about. And what I have boxed is growing degree days, GDD, and cumulative growing degree days. So these are growing conditions. And then the bottom, abundance, CPBA is abundance count. Just below that, next to that red line, are all the genetic data, and they are not significant. They are not predictive. Big disappointment. This is a huge investment in money, of course, to sequence all these beetles, and to get that result was a little bit jarring. I think there's a few reasons for that. One of them you can kind of see in this plot. So the bottom left shows temporal plots of the phenotype, dead beetles, over time. And there are strong spatial and temporal correlations that are evident in that plot. You see the similarity between points is high spatially, and also there's distinct signatures that occur at each time point. This spatial temporal correlation is essentially random error, right? It's something that we'd like to control for while testing our primary fixed effects, which would be the genetic data, the abundance data, and so forth. So we have a really strong signature of spatial temporal correlations that we're modeling well, but it's so noisy, it's so variable that it's overwhelming the information we have in the model to look at the fixed effects. So I think that's part of our problem. That's one thing. The other thing is I think we needed more sampling. We really needed more populations of beetles. So hopefully we can improve as we go forward. So yeah, my conclusion there is that spatial temporal trends and resistance do seem to be predictable by abundance. That's not too surprising. That's essentially effective size and this rate of potential rate of response to selection, as well as growing conditions which just favor large populations. But the genetic data haven't been so useful. Certainly there's other ways to analyze the genetic data that I haven't been able to do yet. And then, this calls out the fact that forecasting, of course, is really hard. Predicting evolution's maybe a fool's errand. But if it can help us improve management practices, even to get some returns, to reduce those insecticides, to get some better control over time, to improve sustainability, this is a worthwhile effort. And it's really not being done. This is not something that most agricultural production regions have in mind. They're not trying to use new technologies to improve outcomes. So I'm hoping that we'll build up a set of tools and ideas that will then eventually translate to more effective management of these environments. Okay, I'm going to end there. Hopefully it's time for questions. I do want to thank many people. The most recent part of my work in the end here, supported by Nur Sultan and Francisco, two students that just graduated. Emma Terrace is a current grad student that's doing a fair bit of this work with me, and Russ Groves has been my longtime collaborator. There have been a whole bunch of other folks that have contributed over time and a bunch of funding sources, so I'm very appreciative of them. And really, I thank you so much for your time, and I hope I can take a few questions here. Thanks.
So I know that cytochrome P450s are also very relevant in the other insect pests, but like other different crops like Amyloids, Transitella, like enable orangeworm for almonds. So I guess, in your opinion, is looking at how very different insects are also upregulating their cytochrome P450s something that'd be like effective for this kind of research, or would it be kind of like compounding?
That's great. So the question is about cytochrome P450s as an example of a suite of genes that were found to be involved in resistance in other insects. I think there's a need, and there's been some efforts to kind of synthesize what we found across insect pests, the kinds of genes that are involved. Commonly, cytochrome P450s are involved. So they're monooxygenases. They kind of are one of the first steps in breaking down that toxic chemical and allowing it to then be sequestered and secreted by the organism. So we see it occur many different species, not always the same family of cytochrome P450s, not always the same cytochrome P450s. They seem to be pretty versatile, but nonetheless, because we know that, that allows us to think about ways to work around that, or at least to track the evolution of that resistance trait.
When you were showing the population structure for introducing the kind of different populations, and you were saying the Northwest population is not really a big pest, is that because it can easily be, or it's more easily managed because the insecticides, or is it not? It's not a problem on it.
Yeah, excellent question. So this is a conundrum to us too, and really interesting. We've tried to study it a little bit because of that. I'd say there are a couple of things. One is it does seem to be a separate establishment of the pests from that ancestor in the plains. It doesn't seem to be connected through demography to those other populations that went east. So I think it has a bit of a different evolutionary history. That's one thing. It also has less genetic diversity, so may not have the substrate for as much evolution. And the West is just a little bit different in how they grow potatoes. They end up having these large circular fields that are more distributed in space. So it's harder for beetles to get from field to field, and it's very dry there. And these beetles are not, I mean, it's not that they can't live in dry places, but they're not typically found in deserty spots. So moving and kind of maintaining population size across those rotating fields, I think is not as easy for them. So there might be a couple of factors, ecological and then inherently genetic that limit their capacity to rapidly evolve. They do have resistance that's developed to neonicotinoids. It's just kind of spotty right now, so we're kind of keeping track of that and trying to understand what they're doing and genetically. But yeah, good question. Siwan, yeah.
Really cool talk.
Thanks.
I was really interested in this cross insecticide resistance and how that evolved. So, to your best knowledge, do you know like how narrow a list of genes are related to that evolved ability, probably metabolic detoxification to multiple different?
Yeah, we had some recent work that's not published, and we're like Emma's working on. She's kind of tracked the gene expression of a panel of metabolic detoxification genes as well as some target sites, and tried to understand in a bunch of different populations across the US, do they share similar genes? They don't. That is to say, like a cytochrome P451 population might be important, but another it's not. So the classes of genes, the pathways are often the same, but the specific genes are not always the same. And I think we need a lot more careful work to really understand it. It's a little hard in the ways we've approached it, like in the kinds of comparisons we've made, to really get at the response to insecticide treatment, what genes are really critical in that. So it's a bit rough what we know right now, but I would say our evidence doesn't point to like a core set of genes that's in all those populations that's like kind of causing resistance. Yeah.
Right. Really intrigued by your idea that there is a lot of standard genetic variation. Because naively, I would have thought the mutational target size would be relatively small, but maybe that's not true. And I guess the question I wanted to ask you is, you talked about some very nice population genetic approaches, which I like. But have you or others thought about quantitative genetic approaches? Is that something that has been done in terms of mapping or even just thinking about variance components of these traits to get more at that standard variation part?
There's a classic study by Dave Hawthorne that looked at KDR knockdown resistance to, I forget what the insecticide was. I think it's organophosphates. Anyway, did a quantitative genetic study in mapping, kind of QTL mapping, and found multiple genes that were part of that, but this key gene, this major effect gene, the KDR knockdown. So that's been done, and really that never was repeated. No other people have tried to do that in this system. They've done in other insects, that are insecticide resistant. We started one to look at neonicotinoids. I had a grad student start that. We kind of got down the road, and we just never quite finished it. We still have the data, and I've been looking at that, but it's not finished yet. So I think, kind of combining these GWAS approaches, bringing in, signatures of selection, the temporal sequencing, like all this is sort of my way of puzzling this out. So I'm hopeful that we will contribute to that at some point. We haven't done that. There. Yeah.
I was wondering, you mentioned that these beetles were like natively present in the Americas, like growing on grass of some sort. So I was wondering if you, I guess, have any thoughts about what drove the transition from like the hurricanes that they made and whether their abundance or population size is different in their grass environment versus
so they live on this weedy plant. It is a solanaceous plant. It's called buffalo burr. It has burrs on it that are these crazy things that get stuck to cattle and stuff. And it's kind of common in where there's a bit of water in the desert or on the field edges of pastures, things like that. It's sort of a weedy species. So that's their ancestral host. But this has been a question of a lot of interest. So how do host shifts occur? What is the host range of a species? And then I can get into the genetics of it a little bit. So what's been studied is not just potato beetles, but different populations of potato beetles, as well as other leptinotarsus species that are not pests. And there are differences in host range. So potato beetles have a pretty broad host range relative to some of their ancestors. So they seem to be already a bit of a generalist. So they may have a predisposition for trying out different hosts. And when we say that, that means we usually test them. Can they eat and survive and reproduce on a host? So that means that they're able to do all of those things. They will eat a little bit of everything, but maybe not be able to survive on it. So how you do those experiments matter. How they do that, I mean, like how they recognize a host, this plant is suitable and that is not, still is a real challenge with insects. We're still kind of understanding their olfactory chemosensory systems. We did a bit of work on this in Colorado potato beetle. We know that they smell green leaf volatiles. So as a leaf is sort of crushed up, the volatile chemicals are in the air and they can smell that and it's attractive to them. So they'll orient themselves and go towards that. And then once they reach the plant, they chew on it and they taste the plant's chemicals and decide whether they want to keep feeding or not. And there are whole suites of genes that are involved in these steps. So smelling, tasting, and it's not just like a one-to-one thing. It's like actually amounts of chemicals and how they're influence the feeding behavior. So it's really pretty complicated and cool. So switching onto that host, we don't really know specifically what genes. We tried to ask this question, but we didn't actually get a good answer. We don't really know how complicated that was. But potato beetles will feed not just on potatoes. They do occasionally feed on tomatoes. They do occasionally feed on peppers, other things. And what's interesting is it varies geographically. So the population in Wisconsin might never eat bell peppers, but the one in North Carolina does. And whether that's inherently due to the genetic variation that's there, or maybe possibly the microbiome that's also been linked to how they break down plants, we don't really understand that yet. So that's a cool area of interest. Is that most of your question, or okay? Yeah. Thank you. Yeah. Cool.
So, I'm wondering about why the potato beetle is so rapid at developing resistance. And if it's standing genetic variation that's the source of resistance, is there a correlation between the levels of variation in these different pest populations and the rates of resistance?
Yeah. This is where I've claimed in the literature, in my publications, that this is the answer. And we did a study. We sequenced genomes of the other species, and we looked for rates of evolution relative to the other species in the genus. And they are elevated. Heterozygosity is elevated. So we find that that there's some evidence for this. But I will say, I have a colleague that I work with who's really interested in other explanations, especially epigenetic mechanisms of evolving resistance. And I painted a story, but there are other avenues we haven't explored. So we don't, I wouldn't say like we've ruled out all the alternatives.
Are there examples of pests that have very high levels of variation, but are slow at developing resistance?
Sure, yeah. And some that develop it even faster. Yeah, there's some
It's not just like one universal.
No. Probably the other best example, or it's Tetranicus is the spider mite, pest around the world, especially in greenhouses and things, resistant to all sorts of things. And it kind of like the potato beetle. I mean, I think one of the parts of the story is that it's that redundancy in the genome. It's like having many members of a gene family that could be at play to solve a problem. And it's the same way. It has a pretty enriched genome in the detoxification pathways. So there may be some species that are super pests in part because of that like redundancy across the genome. Yeah.
I guess just like specifically, I know that you're saying you're looking at other leptin tarsal comparatively. Given there's other like genuses of crescimelids like Schoville and Diabrotica that are also pests. At what point, I guess, do you consider something like too genetically different to be informative of how Colorado potato beetle works?
I have wanted to do that work and not necessarily pursued it too heavily. But so the question's about, well, shouldn't we go and compare other groups of beetles or other groups of insects? And the answer is yes. We would like to see, some commonalities emerge. Can we find repeated patterns across these species and how they're doing this? There are other really active labs pursuing this, but in different kinds of pests like Lepidoptera. So May Berenbaum's done a bit of work, and also Megan Fritz is doing a lot of similar research now. So in Europe, the spider mite. So there are other people kind of pursuing these questions in different systems, and some of the same results are being found. So I think we're going to be ripe for a review paper at some point, and that kind of brings it all together. Yeah, Matt.
You mentioned that duplication is kind of an important aspect for metabolic resistance. Those can be really hard to find in short read data. So I was curious how you're incorporating those frameworks in your tests.
Thank you. Good question. We did do one study on structural variation, and we did a long read sequencing for that. We admittedly did it at a time pretty early, and we didn't have a lot of funds. So I think we compared 8 genomes or something like that, but of some key populations. And we found some evidence that structural variation was at play in loci that we identified as under selection. Some interesting transposable element insertions and kind of repeated units of specific genes like in the esterase pathway that kind of improve detoxification. We need more of that, definitely. So we didn't really survey enough. And there is quite a bit of structural variation. In that paper, we kind of looked at the relative ability of those structural variants versus SNPs to sort of associate with selection. And because SNP variation can be near structural variants, sometimes you can measure the signature of a selection by adjacent SNPs, not necessarily the structural variants themselves. So what I mean by saying that is that I showed you a lot of evidence of areas of the genome that are under selection, but I didn't say what those mutations exactly were. And some of them could be structural variants. So, yeah.
I think you should know, but do you have any idea of why it might not have become a pest on closely related, like other nightshade plants? If it can eat them, and it sometimes does, why does it not spread to those with a big pest?
That's a really good question too. Yeah, I don't know the answer to that. It could be the chemistry of some of the plants, like some of them have a lot more glycoalkaloids, things like that. That's what people have thought about, and they've done those experiments to measure the effect of adding glycoalkaloids and things like that. I'm not sure that that's the answer, but they could, that might be part of it. Yeah. So each of these solanaceous plants are called the nightshades because they're toxic, right? And they're really chock full of all sorts of crazy stuff. And the insects that feed on them are pretty impressive and being able to consume those toxins and sequester them, get rid of them. Yeah.
Thank Sean one more time for a
Thanks, folks. Appreciate it.